Hi again, it's Dr. Roz. In this section, I want to briefly just talk about propagation of plants. Now, when this is done commercially, they have to ensure that this is a very sterile environment because they don't want to pass on any viruses or bacteria that might be present within the plant. So in a commercial setting, this would be done under very sterile conditions. If you just want to try this at home by, taking some clippings or cuttings from a house plant that you happen to have, you don't have to worry so much about the sterility because you're not going to be mass producing this, which is what propagation for us is going to mean, right? Mass producing plants from a piece of a single plant. But for your purposes, you might want to just try and make some clippings at home. Right now everything is budding outside. So if you were to take something from the outdoors, or if you have a plant at home that you might want to try this with, and just maintain as much of a sterile or germ-free environment as you possibly can. So when we're talking here about propagation, we're going to look at what we tend to term micro propagation. We know micro, of course, means small, and propagation is going to be the technique where we can produce a large amount of plants from a single plant. And we do this in an environment that we call in vitro. And by in vitro, basically, we're meaning that we're going to do this within a lab setting. Or for you, it would have to be at home, given our circumstances right now. So if a plant is found to have some sort of feature that we want, if we want really ripe tomatoes, or we want watermelon without seeds, or oranges without seeds, or we want a plant that's going to grow a large eggplant or a big cucumber with few seeds in it. All of these are identifiers of things that are very useful to find in a plant. And by useful, I mean for humans, for us to consume or grow or look at. A lot of times, plants that are grown, they're not grown for food purposes, but like take orchids, for example. We're going to micropropagate an orchid so that we can mass consume orchids that maybe are pest resistant or drought resistant, or have some sort of characteristic that we are interested in. It doesn't necessarily have to be as a food source. Right? So we're looking at taking tissues from the plant here, and usually those tissues are going to be taken off the stem at the shoot layer. So remember, we have the shoot apical meristem, which is where we would take those, because this is an area that has the least amount of differentiated cells. So, right, we want our clipping that we take to grow into exactly the plant that we clipped it from, right? We don't want it to differentiate and turn into some other type of cell. Now, these samples that we take in the lab will be put into a very much an agar. Now, an agar, a nutrient agar in this case, an agar is kind of a gel-like substance. We first make it by taking the agar and heating it, and it liquefies once we get it hot enough. Then we pour it into Petri dishes, and then as it cools, that agar will become more gel-like. And this is the way where we want it. So what we're going to do is sterilize the agar, And again, if you're doing this at home, just put it into water, and you'll be good to go with that. So our nutrient agar in the lab is going to have all of the hormones and other nutrients that plant needs in order to grow at a good clip. This is just kind of what micro propagation looks like. We see we have a nice plant growing here. We're going to take a clipping from the stem of the plant. We are going to heat that agar up, right, so that it's nice and sterile. Then we will put our shoot into there, into the agar gel. It begins to grow. It goes through various stages, and eventually that clipping will grow into and produce a plant like the first one that we had, the one that we took the clipping from. And we can do this many, many times. That's where when we talk about micropropagation, we're talking about taking a small piece of a plant, usually from the shoot, taking a small piece, putting it in sterile agar, and then producing many plants from that one small piece. So, the goal here in the lab is, of course, to make sure that we're producing plants that don't have any viruses attached to them. Now, the meristems would not have, while being low in auxin, they're also low in differentiated cells. They're also going to be free of our viruses. So this is what we would choose to reproduce with. Now, the technique that we use in the lab, and this is used in greenhouses all the time, are going to produce a lot of a plant, right? So that's the first thing, right? A lot of a particular plant. It's going to not take a lot of space, right? Because this is small propagation. And it's going to help us produce things that maybe are difficult to produce or particularly rare to produce. And, that really helps us from a commercial standpoint. And at home, it's going to help you because maybe you have a plant that you absolutely love and that's done really well for you. So you're going to want to reproduce that plant. Mother's Day coming up, maybe you want to make a nice little plant for your mother, your grandmother, your aunts, whatever it happens to be. So here's a kind of a look at what we call cryopreservation. And whenever you see that word cryo, we're talking about something that has been frozen. So here's our seed. And if we freeze that seed at a certain point, it'll stop the germination from occurring until we need it a little bit later. So we might take seeds from areas where endangered species are, and we don't intend to grow them right away. But what we will do is freeze them so that later on down the line we can thaw them out and grow whatever that endangered plant might be extinct at that point, but grow that plant again. Very similar to the cryopreservation of sperm and eggs in fertility clinics, right? We're not going to use that sperm or egg at that given time, so it gets frozen so that later on when it's thawed out, it can be used again. So that's all really I want to talk about with propagation, and this is really going to conclude this part of this chapter. The next part on Thursday that we'll look at is going to look at the parts of a flower and how a flower reproduces. And then Friday we'll have a bit of a review, and then Monday will be the test on this particular chapter on plants. And then all we'll have to do is finish up cell reproduction or cell respiration and photosynthesis. And we'll look at those two things, and then we'll be ready for the block exam. That's it. Any questions, just feel free to drop me an email. Thanks, girls.